everyone, welcome back. In this video, we are going to review how to graph using standard error for your AP Bio class. We are going to do a problem walkthrough of the FRQ question number 2 from 2023. So take a minute to pause this video and read through all of the information on this slide. I am not going to read it aloud, and then meet me back here for the problem solve. Okay, jumping right into question number 2B, it asks us to use the information provided in table number 1 to construct an appropriately labeled graph. So let's first make sure we understand what the data in table 1 is. The first thing I always do is I read the title of my table. It's going to tell me what this information is. So table number 1 tells us the average number of mitochondria in plants exposed to normal and elevated levels of carbon dioxide. Next thing I want to do is I want to look at all of my header rows and columns of this table. So looking at my columns, my first column represents species, so I'm noting that I have 6 species. My second column here in the middle gives me the mitochondria number at normal carbon dioxide levels. And then over here on the right, this gives me the number of mitochondria at elevated carbon dioxide levels. Both of those two columns are given per 100 micrometers squared of cell area. And then we are also given the plus or minus 2 standard errors of the mean. Let's take a second to talk about what standard error is and what it represents. So what happens is that in an ideal world, we would be able to study our population. Our population is literally everything we are studying, right? Or looking at or examining. So in this case, it would be every plant. However, it is highly impractical, if not impossible, in almost all cases to study our population. Therefore, it is very common to take a sample, right? So you might look at 100 or 300 or even, 1,000, 10,000, but it's very rare to actually study your entire population. So because of that, what's going to happen is you are going to have natural variation in your data just because you're picking different samples every time. So the standard error gives us an idea of how much we expect our average to vary, right? So for example, this here tells us that we have, for species 1, we have a mean mitochondria number of 1, but we expect that average to vary by plus or minus 0.1 across different samples. So now that we've talked about that, let's go ahead and jump into our graph. So first thing we always want to do is label our axes. So our y-axis is going to be our dependent variable, which is the variable we are measuring. So this, in this case, is going to be our number of mitochondria. Okay, number of mitochondria per 100 micrometers squared. Okay, great. And then our x-axis is going to be our species number. So species number. Okay, good. So now we are comparing 2 types of data. So we have to think about what type of graph would be appropriate. There's a couple of kind of hints. Again, we have 2 groups of data. So that is going to tip me off that I'm probably going to want to use a bar or modified bar graph. And then second thing that's hinting that I should go that direction is that I have the standard error. It's common to use bar graphs when given the standard error. So I have basically decided to use a bar graph to help display this data. So let's go ahead and start it off. On the x-axis, we want to just make sure that our spacing is even. So we have 20 boxes across here and we only have 6 species. So I think it's going to be appropriate to basically separate every species by 1 box. So I'm going to separate them out like that for species 1. So on the left, this is going to be normal. On the right, this is going to be elevated. And then I'm going to skip a box. And then this would be for species 2. And I'm going to skip a box. This will be for species 3 and so on. I'm going to start off with those and then we can go on from there. Next thing I'm going to need is I'm going to need a key, because if I just draw two two boxes, or I'm sorry, two bars, how would my reader know what's what, right? So I have to tell them that. That's the point of a graph, right? A lot of my students get confused with graphing, but the point of a graph is to allow your reader to visualize the information in a way that makes sense. So when you're graphing, you want to make sure you're doing it in a way that makes sense. Okay, so I'm going to go ahead and normally I would put my key over here. I just don't have space, so I'm going to put it along the top here. So I'm going to leave my empty bar for the normal CO2 levels, and this is the mitochondria. Okay, and then I could say mitochondria at normal CO2. My other bar, I'm going to fill in like so, and this is going to be my mitochondria at elevated carbon dioxide. Okay, great. So now let's go ahead and start off our bar graph. So for species number 1, I have a mean of 1 and a standard error of my mean at 0.1. So the height of my bar, I'm going to draw it up to my mean of 1, like so. And then for my standard error, I'm going to go up by this amount from my mean and down by that amount from my mean. So I'm going to go up by 0.1 and I'm going to go down by 0.1. And then I'm simply going to connect that to kind of draw that bar there. Okay, next I'm going to do my elevated CO2 for species 1, which has an average of 1.6. Okay, let's say it's there. This is much harder to do on the computer rather than on paper, so bear with me. And then again, I have the same standard error for my mean of 0.1. So I'm going to go up by 0.1 and down by 0.1 and then connect those with a bar. All right, moving on to species 2, I have a mean of 0.4. So I'm going to go up to 0.4 like so. And I just realized I forgot to fill in my elevated CO2 bar, so I'm going to do that now. Okay, now going back to species 2, and now I have a standard error of my mean of 0.05. So it's supposed to be halfway between those. That's just incredibly hard to do on the computer, and it should also be halfway going down, like so. Now for the elevated CO2, I have a mean of 0.9, so I'm going to go up to 0.9, like so. I'm going to remember to color this one in since it represents my elevated carbon dioxide numbers. And then my standard error is 0.08, so I'm going to go up by 0.08 and then down by 0.08 from my mean. And then I'm going to draw my bar like so. I feel like most of you are probably already getting this at this point, so if you'd like, feel free to skip ahead in the video to question number 2C. For everyone else, feel free to stick around and watch while I continue to finish out this graph. Okay, so for number 3, I have a mean of 0.5, so I'm going to go up to 0.5. All right, and then my error is 0.07, so I'm going to go up by 0.07, which is maybe there, and I'm going to go down by 0.07. All right, and then for my elevated, I have a mean of 0.9, so I'm going to go up to 0.9, like so. Okay, and then I'm going to color this one in since it's for elevated. And then my standard error is 0.1, so I'm going to go up by 0.1, down by 0.1, and connect them. Moving on to species number 4, my normal mean is 0.3, so going up to 0.3, like so. My standard error is 0.03, which is going to be very hard for me to draw here, so bear with me. Let's say that's about like there, and then going down by 0.3, something like that. And then my elevated mean is 0.6, so about there. I'm going to fill this one in since it is my elevated number, and my standard error is 0.06, so a little over halfway up and a little over halfway down, and connect those. Now moving on to species number 5, I have a mean of 0.7, so I'm going to go up to 0.7, like so. All right, and then my standard error is 0.06, so again, a little over halfway, like roughly like that, and then connecting that. Now, my elevated for species 5 has a mean of 1.5, so going up to 1.5 all the way up here, like so. There we go, and coloring this in so my reader knows that this represents the elevated carbon dioxide levels. Now I have a much bigger standard error here. This is going to go up by 0.22 and it's going to go down by 0.22. So roughly something like that. All right, good. Now moving on to my last species, which is species 6. I have a mean of 1.3, which is going to be here for my normal CO2 levels. Okay, and my standard error is 0.15, so up 1.5, so maybe about there, and then down 1.5, maybe about there. Okay, good. And now the elevated CO2 has a mean of 2.4, so that is all the way up here. Okay, there we go. And now let's fill that in since it's my elevated CO2 bar. Okay, good. And again, I have a pretty bigger, well, relative to my other data, I have a standard error of 0.22. So we're going to go up by 0.22, and then we are going to go down by 0.22, something like that. And then we are going to connect the bar like so. So now that we have an appropriate graph here, the second part of the question asks us to determine which species shows a difference in the number of mitochondria between normal and elevated carbon dioxide levels. So when we look at all of our species show a difference in the height of the bar, right? So that tells us that for all species, we see a difference in the number of mitochondria between the normal and elevated levels of carbon dioxide. So for question number 2C, it says, based on the data in table 1, describe the relationship between the level of carbon dioxide and the average number of mitochondria per unit area of a cell. So again, I'm looking at the normal carbon dioxide versus the elevated carbon dioxide numbers, and I'm looking at the average, right, or the mean. So I'm looking at all of these numbers, and then I am comparing them to all of these numbers here. So what am I noticing? I'm noticing that in every case, the mitochondria when carbon dioxide is elevated is greater than the mitochondria when carbon dioxide is normal. So in order to answer this, I could simply state that. I could say the average number of mitochondria is greater when carbon dioxide is elevated than when carbon dioxide is normal. Thanks so much for watching, and I'll see you in the next video.